today for our first in the neurodegeneration webinar series that's going to be hosted by the UK Dementia Research Institute. Before we start, I'm just going to give a little bit of background to the UK Dementia Research Institute for those who might not be so familiar with it. So if we could just go to the next slide. Thank you. So the UK Dementia Research Institute was founded back in 2017 by our Principal Research Council alongside the Alzheimer's Society and Alzheimer's Research UK. Since then, we've also had input from the British Heart Foundation and Parkinson's UK, which has enabled 2 further centres to be set up to look at vascular dementia as well as Parkinson's disease. As I'm sure you're all aware, one of the greatest challenges facing us is how to maintain brain health in an ageing society. And with that in mind, the UK DRI's objective, healthy brain aging for all years and not merely years to life. So if we could just go to the next slide. Thank you. So at the heart of the UK DRI's mission, we are aiming to underlying pathobiology of degenerative conditions. And with this better mechanistic understanding of the causes and drivers of these diseases, we'll then be able to develop new diagnostics, technologies, and treatments with the potential to deliver life-changing solutions for those individuals that are affected by these diseases or at risk of them in the future. Since we were founded in 2017, the UK DRI has grown to now 9 research centres hosted at leading UK universities, hosting 79 labs of group leaders and emerging leaders, with more than 1,000 researchers, clinicians, students, and support staff working to a single mission. Thank you. So I'd like to welcome you again to the first of these new webinar series. So firstly, I'd like to introduce Professor John Hardy, who is the Chair of Molecular Biology of Neurological Disease at the UCL Institute of Neurology and a world-leading neurogeneticist based at the UK DRI at UCL. John's contributions to neurodegenerative diseases have been revolutionary, and in 1991, his team discovered the first mutation directly linked to Alzheimer's disease, which led to the formulation of the very influential amyloid cascade hypothesis. This groundbreaking work laid the foundation for the first disease-modifying treatments, and his exceptional contributions to science have been recognised by numerous awards, including the Breakthrough Prize in Life Sciences, the Brain Prize, and his election as a Fellow to the Royal Society. In 2022, John received a knighthood for his services to human health and dementia research, and John is now also serving as the Vice President to Alzheimer's Research UK. Our second speaker is Professor Valentina Escott-Price, who is based at the UK DRI at Cardiff University, where she is a professor in biostatistics and bioinformatics. Valentina's academic journey began with a strong foundation in mathematics from the St. Petersburg University before she did a PhD in statistics at Cardiff University. Her pioneering work in statistical methodology has transformed our understanding of the genetic architecture of neurodegenerative diseases. Key member of major international consortia, including the International Genomics of Alzheimer's Project and European Alzheimer's Disease DNA Biobank, Valentina has played a crucial role in identifying new genetic risk factors for Alzheimer's disease, but her expertise stretches beyond this to multiple neurodegenerative disorders. Valentina's work focuses on developing sophisticated new methods to identify individuals at high risk of these diseases using polygenic risk scores, biomarkers, and environmental factors. More recently, she has been leveraging artificial intelligence and machine learning to enhance disease prediction and patient stratification in large population datasets. I'm Dr. Natalie Connor-Robson. I am an AI UK Senior Research Fellow and UK DRI Emerging Leader at Cardiff University DRI, and I'm your chair for today. My lab looks at endocytic dysfunction and Alzheimer's disease, and previously I've worked in the Parkinson's field looking at endolysosomal biology. So like I said, I'm going to kick us off with the talks, but please do put your questions into the Q&A box throughout. Okay, over to John.
Thanks very much, Natalie, and thanks for that generous introduction. I think I'm going to switch my video off because it occasionally cut out there, so I'm not that enamored of looking at my own face anymore, so I'll just switch the video off so it's a bit smoother. So this lecture derives from a series of conversations and some work that Valentina and I have been doing over the last few years trying to interpret GWAS beyond just listing genes. So that's the background to this. And there was a paper put into the chat at the beginning, which I'm sure you can see in your Zoom link, which we'll perhaps go back to at the end. Our aim in today's talk is to suggest a general framework for interpreting genetic analyses, and our intention is to aid in our understanding of the pathogenic mechanisms and also to discuss disease prediction. One of the things that I think is becoming increasingly important with anti-amyloid therapies is to start to understand co-pathologies. And we'll also talk a little bit about how we might approach understanding co-pathologies at the end. Next slide. So I'll just very quickly do the pathologies. Here is the pathology of Alzheimer's disease: large ventricles top left, smaller hippocampus top right at the bottom of that section, tangles bottom left, plaques which are about 1/10 of a millimeter across bottom center, and amyloid angiopathy, which of course is underlying the ARIA complication of therapy, bottom right. Now, when you look at this pathology, you want to understand the order. It's the order of pathology. And that's why I think that's why our finding of genetic mutations in amyloid mutations was important, because it said that at least in those families, amyloid was where it started. Next slide. Here is the pathology of Parkinson's disease, a Parkinson brain with the rather pale nigra top left, a control brain top right with the very dark substantia nigra, and then bottom right is the Lewy body in the substantia nigra, which I'm going to show you an EM of later on with the synuclein protein. Next slide. And here is one of the tangled diseases. This is PSP, and there you can see at the top of the brain, top left, you can see there's some shrinkage of the brain, and bottom right you can see a tangle, a cortical tangle made up largely, in this case, of 4 repeat tau. So this is the pathology. These are the diseases, the classes of diseases we're going to be talking about. Next slide. Now, a paper that to me was extremely influential is the paper at bottom right from Michela Vendruscola at the DRI in Cambridge. And he and his colleagues reported that all of the proteins which we see deposited in that I've just shown you were very close to their, what one might call, crystallization threshold. And he reported this based upon protein chemistry work. But what the other 3 headings there show is that top left, synuclein gene duplications cause Parkinson's disease, tau gene duplications cause tangle disease, and amyloid gene duplications, bottom left, cause Alzheimer's disease. Why is this important? It's important because it gives us one very simple but actually fundamental piece of understanding, and that is that all of these proteins are close to their deposition threshold. So the implication is that by increasing the amount of those proteins very slightly, you can make deposition inevitable. And these very simple cases are gene duplications. They're very rare, but the implication is that the deficits in clearance might lead to deposition. So in terms of my part of this talk, that is really the simple message. Deficits in clearance, we can already predict from these papers, might lead to deposition. Next slide. Now, when we look at the other causes that are known from Mendelian disease, the gene duplications I've just mentioned, the APP and presenilin mutations, which we know about, they increase the amount of A-beta 42 or A-beta 43. These are less soluble A-beta forms, and the tau mutations, which are actually the most common in the UK, increase the amount of 4 repeat tau. So they are also, if you like, increasing expression. And then there's normal variability at the tau gene and the synuclein locus, which predispose to the diseases, the relevant diseases. The tau normal variability at the tau locus predisposes to tangle diseases, and normal variability at the synuclein locus predisposed to Parkinson's disease. So again, the simple view is that marginally greater production contributes to the risk of disease, and a corollary of this is that failures in clearance can have the same effect. Next slide. Now, when we look at clearance, well, I think the biggest thing that the genetic analysis, which we're going to talk about, has taught us is that in Alzheimer's disease, most of the loci we find are microglia, most of them are involved in lipid metabolism, and most of them are a partial loss of function. So that is the set of characteristic which is true for most of the Alzheimer risk genes. When we look at Parkinson's disease, most of them are involved in either autophagy or mitophagy, and again, most of them are involved, most of them involve reduced or loss of function. Rather simple concepts. In tangle diseases, our data is much less clear, and the reason it's much less clear is because the GWAS for Alzheimer's disease are very large, the GWAS for Parkinson's disease are very large, but the GWAS for tangle diseases, such as progressive supranuclear palsy or corticobasal degeneration, are much smaller, and so the number of loci we have found is correspondingly less. But what they do suggest so far is the ubiquitin proteasome. Now, all three of these, microglia, lysosomes in autophagy or mitophagy, and the ubiquitin proteasome, they're all involved in clearance, damage clearance. Next slide. So let's go over the genes very quickly. Top right is one of the earlier GWAS. What's true of this GWAS is still true. And there was a really, again, an influential paper from Leslie Jones, who sadly died a couple of years ago, which to me also was extremely important in focusing our understanding. And she said that most of the genes involve the immune system, and in the context of the brain, that's microglia usually, or she wrote cholesterol metabolism. I would actually change that slightly and say lipid metabolism. I might say, just as we have this slide up here, that this, to all of us, I think, certainly to me, was a surprise. We had all expected to find APP metabolism genes, and we didn't. And I'm going to talk about that next. Now, we had started to do, well, at this time, exome sequencing. We had done a series, we'd started to exome sequence a series of Alzheimer cases that we had collected, and we were interested when we started to see the data in a gene called TREM2. So we contacted all of our collaborators, the guys at Mayo and the guys at WashU included amongst that group, and we found that TREM2 mutations were increased in Alzheimer's disease. And the paper on the bottom, on the right-hand side, is the header from that paper. The guys at Decode identified the same variant, the same major variant, not all of them, but the same major variant as we had done at the same time. But actually, we also, in this paper, had another finding, which we expanded upon later, which was that TREM2 not only was a risk gene for Alzheimer's disease, but was also the gene whose expression was upregulated the most in response to amyloid pathology. So we had two independent findings which happened in the lab at the same time, but in different people. And that was that not only was TREM2 a risk gene, but it was also the gene whose expression changed the most in response to Alzheimer's disease. Amyloid pathology specifically. Next slide. And that led Dervis in my group, working directly with Valentina, to look very carefully at all of the genes which were upregulated in response to amyloid. And you can see that diagram on the right-hand side, and the closer to the center of the diagram the gene is, the more it is upregulated. And there, right in the middle, you can see TREM2, and all the others were genes which had previously been associated with Alzheimer's disease, and so that therefore they were therefore part of the module of expression changes which overall were associated with Alzheimer's disease. So we started to say not just that TREM2 variability was associated with Alzheimer's disease, but that genetic variability in amyloid response was associated with Alzheimer's disease. And then in statistical analysis, Dervis and Valentina identified the genes underlined in blue, which also showed association with Alzheimer's disease. For those of you who are statisticians, what we're actually doing here is in effect reducing the Bonferroni correction by only looking at the genes which are associated with increased expression in response to amyloid. I'll just mention one of the genes, and that is the gene OAS1, which Dervis has continued to work with, and which is part of the interferon pathway. And I mention it also because it's all the same gene is associated with bad COVID. And that's something, again, that Dervis is following up on. Next slide. Now, when we got this, I started to think in particular about how this fitted with Alzheimer pathogenesis. As I've said, we expected to find things responsive to APP metabolism, and that's not what we found. And we found these lipid genes. How can this all fit together? These two papers are what influenced me. TREM2 was a phospholipid receptor, and that was work from Marcus Colonna. And the paper at the bottom, ABCA7, is a phospholipid transporter. So this is pointing at phospholipids, and this was very surprising to me, and I felt it needed to be accommodated with the work in early onset disease showing amyloid metabolism was important. Next slide. So I came up with this suggestion about what the sequence of events are. Could you just press this once? So this is how amyloid metabolism is usually drawn, with beta secretase cutting at the top, and then presenilin gamma secretase cutting at the bottom, and then amyloid going off into the extracellular space. Anyone who works with amyloid, and the guys at the biomarker factory next door work with it all the time, know that you cannot have amyloid in any environment with a lipophilic surface, because the amyloid simply sticks to the lipophilic surface. So I think this part of the diagram with amyloid going off into the extracellular space is misleading. Next press. Rather, I think the amyloid begins to be deposited within the membrane, and I'll say straight away that I don't know which membrane this is, but I think it starts to disrupt the membrane in the way that that slide shows. Next slide. What that leads to is a damage repair which needs to be carried out. And ApoE's involvement in Alzheimer's disease comes as it is part of the repair process of these damaged membranes, it, if you like, takes out the damaged lipids, and then if that doesn't work, the activated microglia come in and start to clear the amyloid. So this is, in my view, the potential connection between APP processing for early onset disease and amyloid removal in late onset disease. Next slide. Patrick Lewis and I have just had this paper accepted, taking this step one step further, saying that on the right-hand side, the damaged membranes are engulfed by microglia, which then extrude the amyloid fragments after delipidation. And the electron micrograph I show there is actually from an electron micrograph from Alex Rower and E.G. Gray, and derives from about 1989, 1990. Similar micrographs were produced by Wisniewski, and what it shows is amyloid fibrils being extruded from microglia. So fitting again with this idea that the amyloid is produced by microglia, even though APP is largely expressed by neurons. In this context, I think it's worth looking at recent papers from Selina Ray and Charlie Arbour, who've shown that British dementia, the other plaque and tangle disease, is the gene involved is expressed almost entirely in microglia. Next slide. So that is dealing then with the role of microglia, and much faster deal with Parkinson's disease. Most of the Parkinson's disease loci are involved in autophagy or mitophagy. And in general, and this is a bit off the topic of this lecture, the mitophagy genes give a rather benign phenotype, hitting largely the nigra, whereas the autophagy genes, and GBA at the bottom there is the classic, give rise to a more general phenotype, which involves cortical pathology as well. So, but these are pointing at autophagy and mitophagy. Next slide. This is the pathology of a Lewy body I promised you at the beginning. And what you can see here is really consistent with what I said about those genes. Here in a Lewy body, you can see damaged mitochondria and damaged membranes. Again, I want to point out that synuclein is a very highly expressed amphipathic protein. That means it sticks on membranes, and what you see is lots of membranes. So one presumes that these membranes have synuclein attached to them. And other pictures of Lewy bodies have synuclein fibrils clearly evident within them. So this fits with really what I think this is pointing to is a consistency with the genetics. Next slide. And tying synuclein to this are these papers from Dimitri Krenk showing that synuclein is largely metabolized through the lysosome, and that if you inhibit GBA in the lysosome, you increase the expression of synuclein, and if you overexpress synuclein, you inhibit the enzyme. So this is pointing synuclein directly at the lysosome, and it therefore squares the circle, so to speak, between lysosomal genes and synuclein overexpression. Next slide. So this is, of course, what that was dealing with was the Mendelian forms of the disease. And all I'll say with regard to this rather messy slide of the Parkinson GWAS is that every individual case of Parkinson's disease has a mixture of autophagy and mitophagy genes together with immune genes. And this will possibly explain the variability of Parkinson's disease, because everybody has a different mix, if you like, based upon their GWAS of risk. Next slide. much quicker on the tauopathies, because I've said we've got much less data of what comes out when we look at the GWAS for the tangle diseases. Top gene is tau. Second one, and very surprisingly, and actually we don't understand why, is a myelin oligodendroglial gene. We don't understand that. Then the other genes are involved in the unfolded protein response and in the ubiquitin and proteasome pathway. In PSP, it's TRIM11. We have just, it's not been published yet, but we have found with Pick's disease, which is a 3-repeat tauopathy, we find TRIM22. My colleague Will Scotton is just about to publish this Pick's disease finding. So again, ubiquitin pointing at the ubiquitin proteasome with PSP and CBD. Okay, next slide. And Karen Duff group, my colleague here at, in fact, my boss here, I should have said, my boss here at the UCL DRI had previously shown that the proteasome was at least part of the way that tangles were cleared. Next slide. Now, years ago, I'd drawn the diagram on the left for the amyloid hypothesis. And, you know, that I think, you know, that diagram, well, I think it's still got some truth to it, but it's worth pointing out, well, its gross oversimplicity is clear, but it points to the idea that amyloid outside a neuron leads to tangle formation and death inside the neuron. That horizontal line there is suggested to be a neuronal membrane. We've been just discussing, though, that this diagram does not have any cells except neurons on it, and it envisages a direct amyloid effect on neurons. With regard to what I've just been saying, and actually talking to my colleagues here and to, as I said, to Michaela Vandruscola, I have become less enamored of this direct idea. For a start, we've never found any real amyloid receptors on neuronal membranes, and that question mark at the membrane remains unsolved after 20 years. Rather now, I start to think of these as the clearance mechanisms which I've been mentioning, microglia, ubiquitin proteasome and lysosome, as actually being, of course, I've discussed them as if they were separate, but everybody who works on biology knows that no two pathways are entirely separate. And I would now say that if you have a cell a system which is close to being failing, then it only needs another push, as shown here, for example, amyloid buildup to cause ubiquitin proteasome and lysosome issues. And if they're close to failure already, that failure will become catastrophic. And that has been recently analyzed by Michaela Vandruscola, and his paper is on I think it's bioRxiv. Next slide. So now we're going to think, though, about the recent GWAS, both in terms of mechanisms and in terms of prediction. I think there have been two issues. The first issue, which Valentina and I dealt with in the review bottom right, is misdiagnoses. And we're starting to find FTD genes in Alzheimer GWAS. But related to that, and I think this is an important step on the way forward, is co-pathologies. So when we collected our first Alzheimer neuropathology data set, I was very strict about ruling out co-pathologies in the cases we took. So they were very pure, if you like. But mostly, though, as any epidemiologist and neuropathologist knows, most cases have mixed pathology. And that the first paper to start to analyze this is this paper at top right, which I really liked, looking at co-pathologies. Next slide. And one of the points about co-pathologies and about the sequence of events is this work which Valentina led, showing that the ApoE locus was entirely if you like, responsible for amyloid deposition. So amyloid deposition is almost entirely ApoE dependent, whereas the dementia in that context is dependent upon the rest of the GWAS hits, so largely the microglial hits. So this is really telling us that we can dissect the pathology using GWAS. Amyloid is ApoE dependent, and dementia in that context depends largely on the GWAS hits. Next slide. Now looking at this paper again, the GWAS of neuropathology, again, amyloid here is nearly entirely responsible for amyloid deposition. Next slide. When we look at the vascular problems in the context of Alzheimer pathology, the top hit is a hit which coincidentally is also a stroke gene. So again, really consistent with the fact that we're getting real biological insights here. Next slide. And when we look at TDP pathology, that comes out of those cases with T-men and granulin, TDP pathology comes out when we see granulin and T-men 106B GWAS hits. So again, consistent with work we already know in terms of FTD and ALS. So we're definitely getting biological insights from these GWAS. Next slide. And Lewy body disease largely has a high ApoE, a high amyloid load. And here, what are we seeing? We're seeing ApoE as a hit again. And so again, consistent with amyloid pathology. We also, on the left-hand side, see some evidence of GBA. But again, amyloid, ApoE always go together. Next slide. So with that background, I'm sorry I'm a bit late in the handover. Valentina, it's over to you.
Yes, as we know, whenever biologists or geneticists talks about their theories, you can give them a gene name and a trait, and they will create a story for you. And sometimes the story is even better than ChatGPT can create, well, most of the time so far. So we will look at the numbers now. If the numbers confirm what John have just hypothesized, and how we can proceed with genome-wide analysis in order to predict prediction accuracy, how can we take these findings to patients and to people, and how can we actually delineate pathogenic pathways. So, as it was said so far, co-pathologies are very common in individuals with late onset disease. Some loci are associated with different pathologies, and we all know that neurodegenerative diseases are age-dependent. And what it means, so if we consider neurodevelopmental disorders, by age 40, when the sample is 30, whenever samples are collected for genome-wide association studies, people clearly have a disease or don't. With the neurodegenerative disorders, age remains to be a mystery, and as age-related to damage occurs, the clearance systems which were adequate in early life become overwhelmed. So lessons what we learned so far from genome-wide association studies based for neurodegenerative disorders. For GWAS interpretation, we need the disease should be labeled correctly. Most of genome-wide association studies for Alzheimer's disease, in fact, has been genome-wide association studies for dementia, especially the latest ones. I will show you examples why. I will be talking mostly again about Alzheimer's disease here because the data exists more for Alzheimer's disease genetic data than for others, but my kind of example, which I'll show you later, is relevant to, I believe, to all neurodegenerative disorders. Ideally, what we would like is to carry GWAS of pathologically confirmed cases and controls, but these studies are quite small. And also we need to add to our genetic studies biomarker-defined phenotypes. and that will break pathogenesis of disease into component parts. So let's have a look at GWAS again. So that's a GWAS from 2009. It's about 11 to 12,000 people, and that is a Manhattan plot. And so to some people, it's reminded of Manhattan skyline. So going now, what's happening now with genome-wide association studies? What if you not only have Manhattan, but we also have Chicago? What if you also have Miami, Toronto, and sometimes London, not so tall, high peaks, but still there? So we end up with a genome-wide association study, so-called for Alzheimer's disease, with a huge number of genome-wide significant losses. But what happens if you look at heritability estimates, which is picked up by genome-wide association studies? So coming back to pathology-confirmed cases and controls, paper was published in 2010, very first GWASs, we pick up about 40% of SNP-based heritability, of broad sense heritability. So as we know, based on studies from twin studies, broad sense heritability for Alzheimer's disease is about 60 to 80%. And by genome-wide association study on pathology-confirmed cases and controls, we pick up about 40-45%. Then the bigger study become the smaller heritability estimates are reported. And this heritability estimates, it's not due to, or differences, not due to software, because I used exactly the same software to estimate all heritabilities myself. And you can see that the bigger studies are, in particular the last one, the smaller heritability we are picking up. So what could be a cause of those findings? First of all, age-specific analysis or age-specific effects. As I mentioned already to you, as compared to neurodegenerative and neurodevelopmental disorders. So we have two age-dependent variables which complicate the interpretation of current neurodegenerative GWAS. First is that pathogenic substrates of disease change with age, with more mixed pathology at later ages, changing the landscape of pathology depending on different age dependencies of damage. And second, allele frequencies, which are allele-specific, are dependent on mortality rates. So if we look at people with about 60 years of age, we have controls, which is a blue line here. It's allele frequency of E4 allele in controls based upon literature, and this is in cases. And when we look at early GWASs and people at early late onset of Alzheimer's disease, we see odds ratio about 3.4, and this was reported and replicated many times. So if you go to age 90, again, that odds ratio drops, and simply because people who had Alzheimer's disease, they're dying in due course, and by 90, we have less people with E4 alleles, and the odds ratio is smaller. Now, if you mathematically continue these lines, we can, and when we go back to the end of the age span, the effect size could even reverse. So it's simply mathematical effects here. What happens with heritability estimates? It's a paper we published in 2023, looking at heritability estimated different scenarios, different disease prevalence estimates, but nevertheless, the slides are busy. Here we look at the sample where Alzheimer's cases and controls are amyloid confirmed. And you can see all the bars are systematically higher, and this is looking at heritability overall at different age counts, and this is looking at when ApoE is excluded from the model. So we do see quite good heritability estimates, 60%. Now, when we look at Gray's data set, for example, which has cases of 70 years of age, but controls a population based here, they are 55 years of age in average, you can see that generally estimates drops down. Similar effect we can see Rosemap and Mayo data set. Again, they are pathology confirmed, estimates of heritability consistently high, independent of scenario. And this is our UK Biobank data. When we look at first three bars, estimates are quite good, and that is genetics including ApoE region, and this one is excluding ApoE region, simply showing that UK Biobank sample being relatively young. So it's at the moment about 70 years of age, and our Alzheimer's disease cases in UK Biobank appearing about 80, in our 79, so it's a small number of them. Again, we're comparing Alzheimer's cases with relatively young population-based controls. And when we exclude ApoE region, the heritability estimate drops. So heritability estimates and any estimates depend on what sample we analyze and at what age we analyze it. Next question is misdiagnosis versus mixed diagnosis. What are we actually predicting? Why are we still difficult to interpret the polygenic risk score analysis or prediction by polygenic risk scores? First of all, there is no agreement to what model to use, polygenic and oligogenic. And polygenic model includes all genes which are not genome-wide significant, which significance below this horizontal line. An oligogenic model actually includes only the top tip of the iceberg or top associated SNPs. So this model shows consistent average accuracy, 65, 70, 74% marks. And this model shows poor accuracy in young cases and controls and quite good accuracy in all the cases and controls. So the next complication of polygenic risk score, what actually approach to use to estimate polygenic risk score? We can talk about this in a different webinar, but I can tell you all the story about that. And finally, which is important, what actually we predict? Lifetime risk at specific time point, risk age at onset, or specific disease pathology, and that needs to be cleared before you use a polygenic risk score and before you choose GWAS, which you use for disease risk prediction. So just to confirm again that polygenic risk score prediction accuracy in Alzheimer's disease depends on age. We had a little experiment. We have taken a sample from ADNI dataset and we split it into age cases and controls younger than 80 and cases and controls older than 80. And you can see again confirming Johnson my observations. When you look at ApoE only in younger sample, accuracy 74%. When you exclude ApoE, accuracy drops. However, when you look at all the sample, as expected, accuracy using ApoE only drops, but polygenic risk score excluding ApoE actually predicts quite well. And that's again showing the picture here. When we look at cases only, that's ApoE frequency with age. It goes down fast in cases and in controls, as you would expect, and the difference is quite big. This is oligogenic model. You can see that oligogenic values going down with age as well in cases, not at all in controls, they're neutral, but polygenic component with age actually increases its value, and therefore we can see the differences in polygenic risk score, excluding ApoE in older age better, it's better predictive than the others. Of course, next bit is to whatever I've told you so far, and Manhattan plots and Chicago and everything I have shown you from European populations, even in European population, it's quite messy. When we look at other populations, it's quite unsatisfactory. And this is unsatisfactory for three reasons. First of all, it means that the therapies which are based upon genetic knowledge will be completely available, will be incompletely available to other populations. And also, as we identify more variants associated with disease in diverse populations, it will give us a deeper insights into the mechanisms of the disease. And again, different haplotype structures mean that precise localization of pathogenic loci is facilitated by cross-ethnic comparisons. And to give it back to John to draw conclusions, the road ahead. John is not listening. Listening.
Thanks for showing us the road ahead there, Valentina.
Of course.
So what do we need to do? Well, I think we need to have pathology confirmed GWAS. And what do I mean by that? Well, we'll always have fewer samples from pathology, but what we should be doing is cross-referencing these huge clinical GWAS with pathology GWAS and using the huge GWAS, if you like, to reduce our Bonferroni correction to look in the pathology confirmed GWAS. That's the way to start. Of course, we need to do GWAS in non-European populations, and we've been doing some of that in a Chinese population with Hong Kong collaborators, and we've been trying to do this also with Nigerian populations too. We should be looking at the GWAS of age-specific risks as Valentina said, what we have done is really we have avoided looking at age-specific risk, but I think it's very likely, based upon what both she and I said, that there will be different risk profiles by age. And one thing that Valentina has been leading on in collaboration with Henrik and Amanda Hesselgrave's groups is to do a GWAS of fluid biomarkers as well, so that we can try and dissect the pathogenesis into stages. Clearly, as we can all see, and as Valentina has started already to do, is to use big data AI type approaches to try and understand the large amounts of data that we and others are now generating. So that's where we think we're up to, and it'd be great to hear what people have to say.
We've got a few in there already, so I'll start by asking you one from David Dexter. So the question is, a lot of GWAS hits in Parkinson's are in the non-coding region. How does this impact our interpretation of what are the true risk genes?
Yes, that's a bit complicated. And so of course, each locus has to be looked at, and the precise approach is different and dependent upon the case. But to give you one example, Helen Plum-Favreau and Mina Wrighton looked at each Parkinson GWAS and tested each Parkinson GWAS for being in a cell biology system for being part of the pink Parkin. So that was one thing that they did to dissect that, and that's how they identified CANSA1. In other situations, we've looked very carefully, and Mina Wrighton has really led on this, to look at which gene has its expression changed at a locus. And doing this, for example, her group identified a synuclein antisense gene at the synuclein locus and a LRRK2 antisense sequence as being important in modulating the LRRK2 gene. So for each case it's different, but for each case it really requires further investigation, really. And there's no one size fits all, but you just you basically have to look very carefully.
Thank you. We have another question here from Gina Scuria. Hopefully I've pronounced their name right. Is there any correlation between ApoE allelic frequency and that of the allelic frequency of protein clearance genes, either UPS or mitophagy or autophagy?
I've never thought about that. Let me I don't think so. But that's a good question, which I have never thought about. Actually, send me an email about it, and then it gets into my hippocampus, and so I can think about it some more. I will say that the ApoE locus in Africans has a much weaker effect on Alzheimer risk. I know that's not an answer to your question, but it's worth stating.
Excellent. Okay, we have a question here from Siddharth and Chandran. Can you speculate more on the role of oligodendrocyte lineage cells in neurodegenerative diseases? Genetics and pathology of MSA seem to suggest that this is understudied.
Well, I will answer by saying MSA is definitely understudied. I can't answer the question. You'll have to give me a bigger budget to do that, Siddharthan. It is a good question. I don't know why. I really don't know. It is an important question. Of course, I'll just say that there is an MSA meeting here at Queen Square in the next month, and I think you're coming to it, so you should ask the people there. I don't know. I really don't know.
Okay, I think there's one here for Valentina from Arthur Roach. Can we hear some examples of the type of questions that the hub will be helping to answer?
Right. So we were hoping to have more collaboration across DRI centers because I think UK Biobank data, for example, is underused. So collaborations, what it will include. First of all, we'll help you to go through instructions how to connect to the databases. If you do have expertise in your team, then we can help you to start or to train you to understand where data is, how to link it together, how to analyze. If you don't have expertise in your team, we will be happy to run the analysis for you because we already connected to the main large data resources. And yes, and that will be on collaborative basis. Hope I answered your question.
Cool. Okay. Another one here is, what is your view on the role of the glymphatic system in clearing neurotoxic metabolites and protein during sleep?
You know, I've never been a fan of the glymphatic system. The guys in Southampton really have been very. I've seen very negative papers about the glymphatic system, about whether it even exists. Of course, I'm not an expert. I'm trying to remember the pathologist in Southampton who discussed this. But what all I will say is that no component of the glymphatic system, Roxana, thank you. Roy Weller, Roxana has just told me on the chat that it's Roy Weller, but Roxana also. So none of the genes that, for example, aquaporin 4 have ever shown up in a GWAS. Of course, there's other reasons that might be the case, but as you know, geneticists just see what is genetically variable and affects risk, and nothing in the glymphatic system comes out. And actually, Roxana is texting me now, and what I would suggest, and we did this several years ago, we had a debate about the glymphatic system. I think that would be something worth doing again, to be honest, because it has become if you like, an established fact, even though the experts in the field don't necessarily believe it.
Okay, interesting. I had a quick question about thinking about why some cell types in the brain seem to be more sensitive to problems with protein clearance than others. Have you got any insight into why that might be? So, for example, you know, dopaminergic neurons in Parkinson's disease.
Well, dopaminergic neurons in Parkinson's disease is one where I do have an idea, but it's almost the only one that I have an idea for, and that is that dopamine metabolism is extreme. It's heavy chemistry, and it gives oxygen radicals and oxidative damage, especially to mitochondria. So the idea that it's might cause a mitophagy issue, I think fits. So that's the selectivity, I would say, in that case. In more generally, I really don't have an idea. Karen Duff's group's looking very closely and at you know, the susceptibility to tauopathies, and so her group might be the right people to discuss that with. Clearly, there is selectivity. I don't know the basis of it. And does Prof. John?
So another one maybe that Valentina can comment on a little bit here as well. So what can we learn from kind of people who age very well and maybe the centenarians? Is there any evidence that their protein clearance mechanisms are preserved or upregulated? You know, how are they overcoming this buildup of nasty, toxic neurodegenerative proteins?
Well, my limited knowledge about centenarian is that they are genetically different, and I don't know how. And I've heard a talk somewhere, which I liked very much, that when you look at centenarians and their lifestyle, it's for some of them it's quite unhealthy, but it doesn't mean that we should have the same unhealthy lifestyle. Our genetics is different, and the response of our genes is different to all this unhealthy lifestyle. That's what I understand. And I also know that some of centenarians are E4 carriers or even E44 carriers, and they still doing extremely well. But that is my limited knowledge about centenarians. How about you, John? What do you know about them?
I would say to look up papers by Henna Holstege, Holstege. She gave the plenary at AI UK. She runs the Dutch centenarian study, and it is very interesting. And I think it's I really think, I mean, you know, there's some things where you wish you could go back in time and you could start a project. Henry Holden here many years ago suggested we got the Queen, it was that Queen, to when she sent the telegrams complimenting people on 100 being 100 years old, she also sent out a spit tube so we could get DNA samples. So that we never followed that idea of Henry's up, but I think it is something that's valuable to do, and I really do think it's something that we as a group should be thinking of doing, collecting a centenarian cohort.
Okay, that would have been quite the present from the Queen. Okay, I think we have time for maybe one or two last questions. So I'm going to go with this one from the chat. Is there a lot of risk genes identified in GWAS whilst exhibiting, if there is a lot of risk genes identified in GWAS whilst exhibiting individual variability, how is that going to be translated to treatments?
Well, I mean, I'll just take the one that I've been most interested in. The one that I've been most interested is PLCG2, which is where there is a protective variant, which is a marginal hypermorph. And so, you know, I think that it would be interesting to develop treatments which, I hope I get this the right way around, slightly reduce the function of PLCG2. That's one where one can see a way forward, even if it's difficult. So I think it depends on the particular gene. Of course, a lot of programs are looking at TREM2 to try and increase TREM2 function. For example, TREM2 activating antibodies and small molecules which activate the TREM2 receptor. So there are there are clear ways that one might think about using the GWAS hits to move forward, but none of them are easy. I mean, that needs to be said. None of them are easy.
Okay, I'm just one last question that we'll do. So a fairly quick answer because we're getting close to time. So one from Giovanna Lally. So do you think it will be possible to revisit the GWAS studies of dementias, not only AD, and try to improve our understanding of co-pathologies? What will be needed to do this?
Well, I'll answer that very quickly. You know what Zane Yamuktain has done here is she's put the Queen Square Brain Bank online. It's now can be viewed and quantitative pathology looked at. And Hugh Morris is putting the genetic data behind that, if you like. So it will be possible to look at a brain without actually doing an MTA and all that sort of shit and get the genetics behind the data that you quantitate on it. So that type of thing. But what we should be doing is replicating and expanding the GWAS that we showed you the header for from the US brain banks.
Okay, thank you very much. Okay, well, just in the interest of time, because we're now at 5 o'clock, I think we'll just say thank you very much again to Valentina and John for a fantastic talk and also for answering all those questions. Thanks to everyone who has joined us, and I hope that you will be joining us again in the future for another Rethinking Neurodegeneration seminar.